Hi, I'm Adam Muzikant, Chief Business Officer at Flexion Therapeutics. It would have been great to visit Europe for the conference. We would like to thank the Alliance for giving us the opportunity to present again virtually this year. Flexion is a public company, and I will be making forward-looking statements, so I'll refer you to our safe harbor language. For those of you who may not know Flexion, here's a brief overview. First, we are solely focused on advancing innovative and locally delivered therapies for musculoskeletal disorders, starting with osteoarthritis or OA. We believe this area has tremendous unmet medical need, which has been dramatically underserved and has seen little innovation to benefit the millions of patients with these conditions. Flexion developed and is now commercializing an approved drug in the US known as ZILRETTA. ZILRETTA employs our proprietary microsphere technology for treating OA knee pain and provides proven pain relief over at least 12 weeks from a single injection. Clearly, ZILRETTA is not a gene or cell therapy, but we've built deep expertise on the back of its development in the field of locally delivered therapies for musculoskeletal disorders. This brings us to FX201, which is an investigational gene therapy injected into the knee, which in the presence of inflammation causes cells in the joint to produce an anti-inflammatory protein known as interleukin-1 receptor antagonist, or IL-1RA. We advanced FX201 into a first-in-human single-dose escalation clinical trial in knee OA patients at the end of 2019. We've made excellent progress in the study and completed dosing in the low and mid-dose cohorts last year. In February, we announced we have dose escalated to the high-dose cohort and that we've expanded the trial to enroll up to an additional 20 patients in each of the low and mid-dose groups. The encouraging data we've seen so far, including some positive signals of clinical activity, support this expansion to further inform our understanding of dose response, evaluate biological activity of FX201 in the joint, and shape the design of a phase 2 proof of concept trial. We anticipate sharing interim clinical data from the initial low and mid-dose cohorts at a scientific conference in the first half of 2021 with data through week 52 in these cohorts expected by the end of the year. Next in our pipeline is FX301, a novel postoperative pain candidate that consists of a NaV1.7 sodium channel inhibitor formulated for extended release in a thermosensitive hydrogel. FX301 will be administered as a peripheral nerve block and has the potential to provide several days of pain relief while preserving motor function. With the FDA clearing its IND recently, in February, we are on track to enroll patients in a proof of concept bunionectomy trial with FX301 in the first half of this year. Knee OA is by far the largest degenerative joint condition, and the burden of the disease is considerable, with millions of patients needing help with routine needs or personal care. Unfortunately, the age at which patients are diagnosed with knee OA has dropped by nearly 2 decades, and this earlier onset has translated into a very long duration of non-surgical treatment, most of which has limited magnitude or duration of pain and symptom relief. The holy grail for OA is a product that modifies the course of the disease by preventing, stopping, or slowing structural degradation of joint tissues. While many have tried to develop a disease-modifying OA drug or DMOD, no one has yet achieved success. Why has the development of new treatments for OA been so difficult? Let's look at the challenges involved in delivering drugs to the joint, shown here. First, drugs that are given systemically do not efficiently reach the joint space because they are either filtered by the endothelium or must get through the extracellular matrix of the synovium. On the other hand, injecting a drug directly into the joint avoids these constraints, but both large and small molecules rapidly leave the joint, either through a lymphatic system or through small blood vessels. We believe an optimal way to deliver a therapeutic to joint tissues for a sustained period of time is through local delivery of a gene therapy viral vector that enables the long-term expression of the therapeutic in the joint, which could overcome both the bioavailability issues of administering systemic therapies for local joint effects and administering intra-articular therapies that are too rapidly cleared from the joint space. We believe that gene therapy is particularly well suited for local treatment of OA for 2 reasons. First, the time course of OA disease progression is very slow, occurring over many years. With gene therapy, we may be able to maintain expression of the therapeutic in the joint over many years with a single injection. Second, because the joint is a compartment, we believe the use of a viral vector brings significant advantages here. The dose requirements of viral particles per knee that will need to be delivered to transduce the joint tissues and provide efficacious levels of IL-1RA in the joint are orders of magnitude lower than those typically administered with systemic gene therapies. This has positive implications on cost of goods sold, especially for a large patient population like OA, where the reimbursement environment will likely not support a 7-figure price for a therapy like FX201. Let's not forget, we also expect limited systemic exposure following local injection, which is supported by our non-clinical data. We believe this benefit of local therapy will lower the risk of systemic safety liabilities. What you see here on the left is the FX201 viral vector map, which has a large genomic carrying capacity. You may be wondering why we're using a helper-dependent antivirus as our viral vector of choice, rather than a more commonly used viral vector, such as AAV. It turns out that for intra-articular injections, non-clinical data suggests that helper-dependent antiviral vectors have a 10-fold higher transduction efficiency compared to AAV, which we believe could have important cost of goods sold implications. FX201 also includes an inflammation-responsive promoter, which turns on IL-1 array expression when the joint is inflamed, providing what we believe will be on-demand anti-inflammatory activity in the OA joint. Let's talk about IL-1RA as a potential therapeutic agent for OA. Well, it's known that IL-1RA is a validated anti-inflammatory protein drug. In fact, anakinra is a human recombinant form of IL-1RA that was approved by FDA in 2001 for rheumatoid arthritis and administered by daily subcutaneous injections. It has a well-characterized safety profile. In many preclinical species studies in OA across species with different models and delivered by both recombinant protein and viral vectors, IL-1RA has been shown to provide both symptomatic relief and slow structural progression, and the effects are consistent and reproducible. Further validating IL-1RA as a potential strategy in OA is that in clinical studies, IL-1RA injected locally has been shown to provide acute pain relief and functional improvements in OA patients. However, these benefits wane quickly, which may be due to anakinra not having sufficient residence time in the joint to achieve a durable effect. Interestingly, last year, some new clinical data emerged from a large CV outcomes trial by Novartis called Cantos that appear to support the hypothesis that durable IL-1 inhibition may result in a slowing of OA disease progression. In Cantos, more than 10,000 patients with a history of myocardial infarction and elevated levels of an inflammation marker called high-sensitivity CRP received injections of the anti-IL-1 beta monoclonal antibody canakinumab or placebo every 3 months for up to 5 years. As part of Cantos, the number of total hip or knee replacements was captured, which provided a unique opportunity to explore the effects of an IL-1 targeted therapy on incidence rates of total joint replacements. What you see here are the cumulative incidence of total hip or knee replacements in those who received canakinumab compared with placebo, representing over 1,300 patients with a history of OA at baseline. Importantly, what the investigators found is that the combined incidence rates for joint replacement were 40 to 47% lower with canakinumab than with placebo over an average follow-up of 4 years. Because Cantos used elevated high-sensitivity CRP levels as an entry criterion, these data suggest there may be a population of OA patients with inflammation that could be responsive to durable IL-1 inhibition as a disease-modifying approach. Now let's look at some data supporting the choice of helper-dependent antivirus as the viral vector to deliver IL-1RA in FX201. In the top panel, bioluminescence data from the mouse shows how the helper-dependent antivirus stays local to the joints 3 days after intra-articular injection, suggesting a potential reduced risk of systemic toxicity given the lack of biodistribution outside the joint. In the bottom panel, you can see that the helper-dependent antivirus maintains expression of the luciferase gene in mouse after intra-articular injection, as shown in the dark green curve, compared to the rapid degradation of the first-generation antivirus, shown in the light green curve. The helper-dependent antivirus maintains expression out to at least a year in the mouse model, which was the duration of the study. One of the attractive elements of FX201 is the inclusion of the NF-kappa B inflammation-responsive promoter to control IL-1RA production. We included this data to demonstrate how this inflammation-responsive promoter in FX201 works. 4 different dose levels of the equine equivalent of FX201 were injected into joints in a single normal horse, and IL-1RA levels in synovial fluid were measured over time. As you can see in the graph, there is an immediate increase in IL-1RA on day 1. These high levels indicate that the vector itself may have triggered a transient acute inflammatory reaction, which in turn activated FX201-mediated IL-1RA production. Then, on days 2 to 5, when NSAIDs were given to suppress inflammation, the expression of IL-1RA was reduced and stayed low from after day 21 until 3 months after dosing. On day 92, the horse was injected locally with LPS, a pro-inflammatory agent, and as you can see, this turned on the expression of IL-1RA in the joint. Following the study in normal horse, the gold standard OA model, was employed to study both symptomatic and potential disease-modifying effects of the equine equivalent of FX201. In this horse model, a fragment is created in the joint, and horses are exercised for several days to mimic natural OA conditions. What you see here are the compelling efficacy data from the equine OA model with FX201. On the left are the scores for lameness, range of motion, flexion, and effusion for the 2 different doses of FX201 compared to placebo. And on the right are the structural changes as assessed by both gross and microscopic scoring at the end of the study. For both doses, you can see significant improvement as evidenced by the lower scores compared to placebo in each of the 4 pain and function parameters, as well as significant improvement in the joint structure, both at the gross level and microscopic level. These data are particularly striking given the study had just 4 horses per group. To support the filing of an IND, we conducted a full non-clinical pharmacology and toxicology program. What you see on this slide are further data supporting potential for disease modification from a rat ACL tear model of OA. In this study, 2 different doses of the rat equivalent of FX201 and vehicle were injected into a rat knee at 7 days after injury. On the left are histology sections at 12 weeks after injury from joints treated with vehicle or FX201 at 2 different doses, one of which was equivalent to the clinical starting dose in our phase 1 NEOA trial. With vehicle treatment, there is clear structural damage, as indicated with the black arrow, highlighting fissuring of the cartilage surface and substantial remodeling of subchondral bone, as shown with the black asterisk. Upon treatment with both doses of FX201 in contrast, the cartilage and bone damage were mitigated in a dose-dependent fashion. The full data are shown on the right, where the cartilage and bone damage scores increased with vehicle and will return to almost normal levels with FX201 treatment. Of course, safety and tolerability are critically important elements to assess as we moved FX201 towards the clinic, and our team put together a strong non-clinical data package which supported the successful opening of a US IND. Together with the solid efficacy that FX201 has shown in multiple animal models of OA, at doses equivalent to those now being studied in the clinic, results from our non-clinical safety studies support a potentially large safety margin in the clinic as well. Importantly, in our GLP biodistribution study, we found very limited distribution of the FX201 vector outside the joint, as assessed at multiple time points after injection, and no shedding. To touch briefly on manufacturing, we have been collaborating with SAFC in Carlsbad, part of Millibor Sigma, to produce the initial supply of FX201 clinical trial material. We've been successful using the current process under GMP and have shown good yields with low levels of helper virus. Our in-house biologics team is now working on scale-up and other activities in preparation for post-phase 2 studies. To give you a flavor for the ongoing first-in-human trial with FX201, it is a single ascending dose study with 3 dose cohorts in NEOA patients, where the primary objective is to assess safety and tolerability. However, given that FX201 is being studied in patients, we have also included a number of exploratory endpoints listed here to assess potential clinical and biological activity of FX201, explore their potential relationship to such clinical parameters as HS, CRP, and also evaluate any immunological response to the vector. Last, let's summarize what key milestones have been achieved for FX201, where we are now, and where the program is headed. Our interactions with FDA and EU regulators on FX201 have been encouraging. We have broad worldwide IP protection on FX201 into the year 2033, and just last fall filed a new patent application for FX201 covering composition of matter and effective dosages in the treatment of OA in humans, which is expected to provide protection until 2039. We successfully opened a US IND and initiated our first in-human single ascending dose clinical study of FX201 in NEOA patients at the end of 2019. Importantly, we were very pleased to announce in February that we dose escalated to the high dose cohort and expanded the trial to enroll up to an additional 20 patients in each of the low and mid dose treatment groups. The encouraging clinical data we've seen so far support this expansion to further inform our understanding of dose response, evaluate biological activity of FX201 in the knee, and shape the design of a phase 2 proof of concept trial. We have initiated treatment of patients in both the expansion groups as well as the high dose cohort. We anticipate sharing interim clinical data from the initial low and mid dose cohorts in the first half of this year at a scientific conference, and are quite excited about our progress with the program. We believe FX201 has substantial potential to provide durable pain and symptom relief, and possibly arrest disease progression in NEOA patients. Thank you.